I'd like to begin by thanking Charlie for the invitation to present and make a contribution to this meeting. What I'd like to do this afternoon is to hopefully give you a brief overview of the therapeutic potential of the hormone Relaxin and hopefully highlight its potential application to Dupuytren's disease. And I have nothing to disclose. So Relaxin is a 6 kilodalton dimeric peptide hormone, which is structurally related to the insulin family of peptides, and is mainly produced by the corpus luteum within the ovary of pregnant mammals, and to a lesser extent in the prostate and or testes of males, and has more recently been identified in the heart, lung, and kidney and brain of both genders. The mature hormone, which is shown here, is composed of a two-chain structure that is linked by disulfide bonds in a similar manner to that of insulin, whereas 24 to 25 amino acid long A chain maintains the stability of the molecule, while its receptor binding domain is located in its 29 amino acid long B chain. Humans and higher primates actually contain 3 relaxin peptides, and in humans these are termed H1, H2, and H3 relaxin respectively, while most other species, including rodents, only contain 2 of these peptides, and these are termed relaxin, the species equivalent of H2 relaxin, and relaxin 3, the equivalent of H3 relaxin. Now H2 relaxin and its species equivalent peptide represent the major stored and circulating forms of relaxin in their respective species, and will be the forms of relaxin that I'll be focusing on in my presentation today. Several studies have now shown that H2 relaxin has a number of beneficial and potentially therapeutic actions in the body, and these are based on its well-established antifibrotic, anti-inflammatory, anti-apoptotic, pro-angiogenic, and vasodilatory actions. And these have been shown to be mediated through its leucine-rich repeat-containing G-protein coupled receptor known as relaxin family peptide receptor 1 or RXFP1. Now, I just wanted to briefly highlight that based on the vasodilatory effects of relaxin, which have been demonstrated in a number of vascular beds in several organs, H2 relaxin is currently in clinical trials for acute heart failure, where phase 1 and 2 of these trials have recently been completed in over 250 patients, and have demonstrated that a 24 to 48 hour infusion of the peptide was not only well tolerated, but was able to improve dyspnea, cardiac output, and other measures of cardiac and renal function. While the number of rehospitalizations and deaths in patients receiving H2 relaxin in a 3 to 6 month follow-up period was much lower, as shown by the several green lines here, compared to those receiving the placebo, as shown by the red line here. And based on this data, an expanded phase 3 trial has recently been commenced on over 2,000 patients with acute heart failure. But for the purposes of this meeting, I'd like to focus on the antifibrotic effects of H2 relaxin, and these have been based on several fibroblast culture models which have been demonstrated, which have used, primarily human and rat fibroblast culture models from organs such as the skin of normal and scleroderma patients, the lung, uterus, kidney, liver, and heart. And these combined studies have demonstrated that relaxin does not affect basal fibroblast function or matrix turnover, but is able to significantly inhibit the influence of several pro-fibrotic cytokines such as TGF-beta1, angiotensin 2, and interleukin-1 beta, and this in turn inhibits the influence of these cytokines on fibroblast proliferation and differentiation into myofibroblasts, and hence the ability of these myofibroblasts to synthesize various matrix proteins, mainly collagen. Now, additionally, relaxin has been shown to promote the expression and activation of various matrix metalloproteinases, mainly focusing on MMPs 1, 2, 9, and 13, while also being able to inhibit the actions of the tissue inhibitors of MMPs known as TIMPs, or at least increase the net ratio of MMPs to TIMPs, and this in turn favors a net increase in collagen degradation. So based on these consistent findings that relaxin can consistently inhibit the actions of TGF-beta, more recent attention has now focused on how relaxin affects TGF-beta signaling and on the SMAD proteins, which are a group of regulatory proteins that are involved in TGF-beta1 signaling. Now, to date, at least 9 distinct SMADs have been characterized, and it is now known that SMADs 2, 3, and 4 actually promote the actions of TGF-beta as the phosphorylation of SMADs 2 and 3, the complexing between SMADs 2, 3, and 4, and the translocation of these complexes from the cytosol to the nucleus to initiate gene regulation are key events required for TGF-beta signaling. On the other hand, SMAD7 has been shown to be antifibrotic as it is able to antagonize the effects of TGF-beta at both the receptor and nuclear level. Using human renal fibroblasts, Strutz and colleagues back in 2005 were able to demonstrate that relaxin was able to specifically inhibit the phosphorylation of SMAD2, which was shown by Western blotting using a specific antibody to SMAD2 phosphorylation, which in turn inhibited the complexing between SMADs 2 and 3 to interfere with TGF-beta signaling of myofibroblast differentiation and MMP activity, without having any direct effects on SMADs 3, 4, and 7. And as an extension of this work, our lab was then able to use renal myofibroblasts from the obstructed kidneys of rats and a mouse model of tubular interstitial scarring in vivo to demonstrate that relaxin is able to mediate its inhibitory actions by signaling through its primary receptor RXFP1 as well as the nitric oxide pathway, specifically via NNOS or NOS1, nitric oxide and cyclic GMP to inhibit the phosphorylation of SMAD2 as a means of interfering with TGF-beta signaling of myofibroblast differentiation and the ability of these cells to synthesize collagen. And this is demonstrated here, where the inhibitory effects of relaxin on SMAD2 phosphorylation in these cells was significantly blocked by the general nitric oxide inhibitors, LN-MMA and L-NAME, the NNOS specific inhibitor, NPLA, and the cyclic GMP inhibitor, ODQ, but not the INOS inhibitor, 1400W. And likewise, the inhibitory effects of relaxin on alpha-SMA expression, which was used as a marker of myofibroblast differentiation, was significantly blocked by both L-NAME and LN-MMA, but not DN-MMA, which is the inactive isoform of NMMA. And again, its inhibitory actions were blocked by the NNOS-specific inhibitor NPLA and the cyclic GMP inhibitor ODQ, but not the INOS inhibitor 1400W. And our current work is now focused on further investigating the interaction between relaxin, nitric oxide, and TGF-beta signaling as a means of controlling the regulation of fibrogenesis. Now, in studies using human dermal fibroblasts from scleroderma patients, relaxin has also been shown to act in synergy with other collagen inhibitory agents, such as interferon gamma, to downregulate the collagen in these cells, where it was found that lower concentrations of H2 relaxin in conjunction with interferon gamma were able to more effectively downregulate the collagen in these cells compared to high concentrations of either therapy alone. So based on these various cell culture studies, the antifibrotic effects of relaxin have also been evaluated in several rodent and larger animal models of injury or disease in vivo. And while I don't have the time to go through each of these models in detail, H2 relaxin has been evaluated in at least 2 rodent models of dermal scarring in vivo, which were caused by fibrous capsule formation around osmotic mini-pump implantation in mice or polyvinyl alcohol sponge implantation in rats. And it was found that as little as 2 weeks of H2 relaxin treatment at concentrations found in pregnant rodents, it was able to dose-dependently decrease the collagen associated with the fibrous capsules, while also inhibiting the Mason trichrome staining of collagen and other matrix proteins within the skin of these animals. Now, H2 relaxin has also been evaluated in several diverse models where 2 to 4 weeks of treatment in the form of osmotic mini-pumps, which allows for the continuous infusion of the peptide into the circulation of these animals, has been evaluated in models of fibrotic cardiomyopathy, in addition to models of ischemic, hypertensive, and diabetic heart disease, in models of renal injury and mass reduction, in addition to hypertensive and obstructed kidney disease, in the carbon tetrachloride-induced model of liver injury, as well as in bleomycin and ovalbumin-induced models of lung injury. And importantly, H2 relaxin has been demonstrated to reduce or reverse the fibrosis associated with every in vivo model so far examined. So H2 relaxin appears to meet several criteria of an effective antifibrotic in that it is able to ameliorate myofibroblast-induced fibrogenesis both in vitro and in vivo. Its effects are species and organ independent and occur with rapid occurring efficacy. It is able to abrogate aberrant matrix deposition in multiple organs with similar pathologies. Its effects are, it is effective even if treatment is administered to established phases of the disease. Its effects are specific to the site of injury. And these several studies have shown that it does not affect basal matrix or collagen turnover, highlighting its safety as a potential therapy. And these various studies have shown at the in vivo level that the antifibrotic effects of relaxin are again mediated through its ability to either inhibit TGF-beta expression itself or the influence of TGF-beta on myofibroblast differentiation and matrix synthesis, while it is also able to promote MMP expression and activity while decreasing the expression of the TIMPs to promote an increase in matrix degradation. Now, consistent with these findings, relaxin gene knockout mice, which lack the major stored and circulating form of mouse relaxin, undergo an age-dependent progression of collagen accumulation in several organs, as shown here, and this results in a progressive increase in dermal thickening associated with increases in types 1 and 3 collagen, compared to that measured in age-matched wild-type mice. But again, this phenotype of increased dermal collagen deposition can be reversed by as little as 2 weeks of exogenous H2 relaxin replacement therapy back into these knockout mice as shown here. And again, its antifibrotic actions were shown to be mediated through its ability to downregulate myofibroblast differentiation compared to that measured in untreated knockout mice and back to levels measured in wild-type control animals. So H2 relaxin appears to be a naturally occurring endogenous regulator of myofibroblast activity and can be added to the list of antagonists of myofibroblast function, which include hepatocyte growth factor, bone morphogenic protein 7, SMAD7, and interferon gamma. And with its ability to regulate TGF-beta signaling and myofibroblast differentiation, may have potential application for Dupuytren's disease. I'd just like to conclude by acknowledging the support of the Relaxin Group at the Florey Institute, which is based at the University of Melbourne, as well as the National Health and Medical Research Council and National Heart Foundation of Australia for the current support of my work. Thank you.